This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 14, recorded on July 29th, 2010. Hey everybody, I'm Vincent Racaniello, and I'm with Dick Despommier.
Hey Vince.
Dickson, I mean.
That's okay, you can be informal.
You told me once you prefer Dickson.
I have no preferences. People tend to call me what they like.
Do they call you late for dinner?
Well, that's the one point that I
You
meant
don't want
to add.
to be called late for dinner.
You can call me anything you want, but don't call me late for dinner. That's so old, Vince.
All right.
That's as old as I am.
Should I cut it out?
No, you can leave it in. People like these old saws.
My jokes are non-existent.
I wouldn't say that at all.
Anyway, how have you been?
I've been well, thank you.
Yes?
Yes, I have.
I saw you this week.
And I'm sorry that I missed our other show, which was held out in Bozeman, Montana.
You were requested. They said, is Dickson here? Is Dickson here? I said, he was here last week, but he's not here this week.
That's actually the truth.
That's all right. Perhaps another time. If not, they will get you vicariously. No, that's not the right word.
No.
They'll get you recorded,
Virtually.
Right? Virtually, yes. Thank you very much.
It's okay.
All right, so last time we finished up toxoplasma.
We did. We didn't quite finish it, but we left that topic for another time.
Oh, we'll come back to all of these. Because
like
we're
to.
doing these forever, right, Dickson?
I hope so.
As long as we're alive.
As long as they continue to request them, we will continue to do them.
You know what? If the email stops, then we can assume no one is listening. And then we'll stop.
That's right.
But we still get emails, so.
Yeah, I'll write you an email. You write me an email.
Yeah, we'll come
keep
back
going.
to Toxo.
We'll just keep going.
So what is on the plate, if you will? Is that the appropriate phrase?
You could say that.
For today.
Well, we're going to continue our journey through the wonderful world of parasitic protozoans.
Protozoans.
And those are single-cell organisms that everybody knows. And we're going to discuss the Leishmania group today.
Leishmania? Like as in I got you on a leash?
Yes. Mania.
Leishmania. I always thought it was Leish. I guess I was
Leish or Leash. I guess if you're Scottish, you'd know the correct pronunciation of this one. But
Leishmania. So
it's
people
named
who,
after a Scotsman by the name of Leishman, or a Leishman, depending on...
So people who work on this are Leishmaniacs.
That's exactly right.
In fact, I think we had an email from a Leishmaniac last time.
Yep.
So that's interesting that you should choose Leishmania.
Well, it's part of the series that we're continuing.
Protozoan. But we had an email from Sophie, who wrote, I recently got a shelter dog from Spain where several horrible dog parasites are quite common. Therefore, I've been reading up on them. But I must admit that I really miss a TWiP covering Leishmania.
Or Leishmania.
I think you've mentioned it before, but that's not enough.
Correct.
Can you imagine we've mentioned talking about it? And it's not enough just to mention it.
No, I think we should devote an entire show to it.
So here you go, Sophie. How many shows do we need for Leishmania?
I think 2. Because there's an old world Leishmania.
Is it mania?
Or Leishmania.
Come on, Dick, let's get it right. Leishmania or Leishmania or Leishmanosis? What is it?
I'm leaving it totally up to the audience.
Well, we're going to get all kinds of email corrections. If we
I used to attend the American Society for Tropical Medicine meetings regularly. In fact, religiously. I didn't miss one during my 29 years of laboratory-based research.
Right.
And I've heard it pronounced both ways by people who are experts in the field. So
both Leishmania and Leishmania.
That's
okay.
Right.
So, folks, there you go. He was at these meetings full of experts, and they pronounce it both ways.
I believe the British would have pronounced it Leishmania, and we would have pronounced it Leishmania.
What do you prefer?
Either or.
I like the idea that it's a mania.
Yeah.
Leishmania.
Yep.
Okay, so we'll do maybe 1 or 2 on Leishmania.
That's right.
Is this as successful a parasite as toxoplasma?
There's nothing more successful than toxoplasma. Nothing.
Equally? Not even equal.
Well, the reason why it's not as successful, Vince, is because of what? You're looking at the page in the book that tells you exactly why it's not as successful.
The vector.
Exactly. It's got a
Dix
vector.
and Doxo didn't have a vector.
That's right, Vince.
Oh, so you're always limited by your vector, huh?
That's the difference.
Wow, I'm really learning here.
Oh, come on, Vince. This is like virology with bigger organisms. That's all it is.
So the vector is limiting Leishmania.
In fact, as we will discuss later, Leishmania have viruses of their own.
Do toxos have viruses? We actually never got into that.
I don't think they have any double-stranded RNA viruses, but we could look it up on the internet on PubMed while we're talking. Yes, they do. And some other protozoans have viruses as well, and they're receiving an increased number of
Leishmania RNA virus. So we have to quote Jonathan Swift again.
I believe that's right. I think the audience is probably tired of hearing that one by now.
All right. Wait, where should we start?
I think we should start with just saying what they are and where they're found and what kinds of them there are.
Do you lecture on this in your course?
No, I don't.
Are you serious?
I'm dead serious.
Why is that?
Because up until very recently, we haven't found Leishmania in this country, in the United States. So I tailored our parasitic disease course to our medical students to only the parasites that they would find in their practice when they got out of school.
Ah, and it's never imported?
It is. I mean, occasionally in the past, when a lot of United States geologists and geochemists and chemical engineers were involved in Saudi Arabia, for instance, for the oil industry, lots of them came back with cutaneous leishmaniasis. Okay, and I've already told you what kind there are now. There's two kinds. There's visceral leishmaniasis and cutaneous leishmaniasis, just two kinds.
And not pulmonary. The visceral can be pulmonary?
I didn't say which viscera. Okay. I just said it's visceral. Okay. So visceral means that the organism somehow gets out of the skin, because that's where the vector deposits it to begin with.
Always.
Always.
All right.
And migrates, not on its own. It gets help. Just like toxo gets help from macrophages, this one gets help from dendritic cells in the skin. And the next thing you know, this organism is found in lots of other places.
It's interesting because dendritic cells, of course, their job is to look for trouble.
Look at that, Vince. Look at that. And they found it. They got more trouble than they bargained for in this one, however.
Same thing with HIV.
Tell me about it.
HIV, when it enters us initially, binds to dendritic cells.
Isn't that interesting?
It doesn't go inside them. It just sticks on the outside. And where do dendritic cells go when they sense trouble? Do you know?
Well, Vince, I think they head off to the bone marrow.
Lymph nodes.
And the lymph nodes.
They go to the lymph nodes.
Okay.
And there they say
to give the messages
to the lymph nodes, they say to the lymphocytes, we have a problem.
That's right.
Houston, we have a problem.
Exactly right.
And what are the hosts for HIV?
Turns out to be B cells.
T cells, CD4 T cells.
Yeah.
T cells, not B cells.
Oh, sorry about that.
CD4 positive T cells, which are in the lymph node.
Right.
So what has this dendritic cell done? It has acted as a Trojan horse, except in a real Trojan horse, the guys are inside.
Yeah.
But here are the viruses. So isn't this brilliant how it's evolved?
It is.
So Leishmania binds a dendritic cell.
Not just binds.
Gets in.
Goes inside. It's actually engulfed. And in an attempt to digest it. And we'll get to that later.
And that brings it to lymph nodes. Okay.
We'll get to that later.
So
so there are two kinds. Cutaneous
and visceral.
And visceral. And then there are two places. New World and Old World.
New World.
So we are living in the New World.
The Old World is Africa.
The Old World is anything not North America or South America or Central America or Mexico. Anything on the other side of the pond, as they would say.
Yeah, the stuff we were discovered by the conquistadors and all those guys, and that's the New World.
Maybe by the Vikings. That's right.
Or maybe before then. Yeah.
Or maybe even before then. That's correct. Yeah. But the real point of this is that what's fascinating, Vince, is that there are two vector groups that transmit this infection.
Old and New World.
Old and New World, and they're both called sandflies. But in the Old World, they're called phlebotomine, okay? And in the New World, they're called Leishmania. So as the continent of South America and Africa split apart during the Jurassic, the split between many of these organisms occurred, so that we'll also discuss later in another show, African versus American trypanosomiasis. Now in that case, they're both vector-borne infections also. In Africa, they're carried by tsetse flies.
Yeah.
In South America and Central America, and even parts of the United States, they're carried by reduvid bugs.
Right.
So the vector species for those two are quite different. But the vector species for Leishmaniasis are quite similar.
So there was a common vector ancestor before the continents separated.
Apparently so.
And
that hasn't actually evolved very differently.
So that when they split the sand, whatever the vector was, then evolved and became different.
That's right. And in fact, Vince, we have sandflies in the continental United States, but we didn't have any Leishmaniasis until very recently. Now, it's a vector-borne infection, you see. If that's the case, then how lucky is that species of Leishmania to have come to this country in a dog and been bitten by our own domestic sandflies and succeeded. And so now we have an endemic cycle of Leishmaniasis.
So we have it now in the U.S.?
Yes, we do.
So you should start teaching it, shouldn't you?
Well, it's for dogs, though, you see.
It's only dogs.
Well, not just... Well, we haven't had any human cases actually reported, as far as I know, for the dog origins of Leishmania tropica, which came across from Middle Europe.
So the vector has been in the U.S. for ages, right? The sandfly.
The vector has been here for as long as we know.
Just like the vector of dengue virus has always been in the U.S.
Sure,
right?
Aedes aegypti.
And
That's
now
right.
only now is it seemed to be in Florida, the disease, and maybe will spread.
Well, the same thing happened with West Nile virus.
West Nile, the vector was always here, right?
That's right.
Culisine mosquitoes?
Culex pipiens. That's the primary vector for that.
Interesting.
And by the way, I should mention to our other listeners that this is the beginning of the West Nile season, and we've already had some human cases, so it's happening as we speak. But in very hot and very dry places only. That's right.
I heard at ASV some
Yes.
cool talks on West Nile. There's a big group up at the public health, and what is it up in Albany?
Oh, yeah, the New York State...
Department of Public Health, I guess, or
Correct.
Department of Health.
Yes.
They have quite a large group, and they do a lot of environmental sampling and trying to understand mosquito movements with respect to viruses, West Nile, and how the viruses change. Cool stuff that I had not been aware of. Laura Kramer is the head of that group. They're doing great stuff.
Perhaps on another show we should get her to come on. So Leishmania then exists not only as visceral versus cutaneous, and not only as Old World versus New World, but there are multiple species in both places.
Some of them called Leishmania something, right?
Well, there is the central visceral Leishmania that was one of the first discovered, although it was known for many, many centuries among people as to the cutaneous form, because it was called various things, but the most common name for it was Baghdad boil.
Wow.
And I've
heard that.
It's a growing lesion that starts out as a little red papule, and then it becomes macular papular, and then it becomes craterform. And the craterform then continues to grow outwards, and the organisms are found at the living edge of this craterform ulcer, and it can be as large as a silver dollar before it resolves. Now, if that happens on your face, you're scarred for life on your face. No matter where it happens, you're scarred for life. So, as the Chinese discovered the cure or the way to intervene with the life cycle for smallpox by scarification, so too did they use the resolving craterform ulcers from cutaneous leishmaniasis to inoculate people with an attenuated form of the organism and to create huge numbers of resistant people as the result. The Russians, in fact, for many, many, many years, were actually touting a vaccine, which was an attenuated Leishmania organism that you could scarify into the skin and get this small lesion to develop, and then a small scar would develop, and it wouldn't go any further than that. So at any rate...
are there strains that only do visceral and only do cutaneous?
Well, we thought so until the Operation Desert Storm, but we're jumping ahead of our story here. Okay. So I was going to tell you about the main one. The one that everybody's afraid of.
Which is which one?
It's named after two people. All right. One guy's name was Donovan, and the other guy's name was Leishman. And they both were operating as British medical officers in India. And at that point, Ronald Ross, soon to become Sir Ronald Ross.
Ronald Ross of the... wait a minute.
Got a Nobel Prize for something, Vince. It was in our...
Malaria.
Yes.
A vector transmission of malaria in birds, though, right?
That's correct. In birds. Boy, you're sharp.
No.
Yes.
I just listened to you, Dickson.
No, but you remember, too. I listened to you, too, but I forget.
How many types of viral genome are there?
7.
Okay. So the same Ross is...
so Ronald Ross is now sitting back in London as the senior editor for the British Medical Journal. In comes these 2 papers from different parts of India. They say the same thing. These are sick people. They're dying. They think it was caused by an organism. This was the age of discovery for microbes in general. And Ross takes a look at the sections, takes a look at the epidemiology expressed by both of these individuals, and decides that these are not 2 different things, but rather they're the same things.
Yeah, you think that would happen nowadays?
I doubt it. So Ross, on his own, of course, decides that he's going to rename the organism, right? He's going to rename it Leishmania for Leishman and Donovani for Donovan.
No, they had different names that they had planned, right?
They had.
Leishman and Donovan.
That's right. They were probably going to name it after the area that they isolated it from in India. But the point is that it became known as Leishmania Donovani.
So what he decided stuck, in other words. He said, if you want to publish this, you have to do this.
Well, he published both of the papers together and said these are the same things in different places. So, but it's a deadly disease, Vince. It's got a high mortality rate and a very high morbidity rate. And it is still around, and there are no vaccines for this one.
How many people a year get this?
In the millions.
Really?
It's in the millions.
Mostly where?
Mostly throughout South Asia and throughout the Middle East. There is some of it now in the New World as well because it's been imported.
South America?
Yeah, that's right. So there's Leishmania Donovani is in a lot of different places now.
And this causes visceral leishmania.
It does.
Where the disease is called leishmanosis?
Leishmaniasis.
Leishmaniasis. Okay.
That's right. And it's a terrible disease that attacks the reticuloendothelial system. It's attacking our attack system. This is our second line, third line of defense. Come on.
Yeah.
What's wrong with this picture? This is a very clever organism in terms of its biology. It's been highly selected for life inside the very cells that we depend upon for our own protection.
So it was discovered in the 1800s?
That's correct. So why don't we talk about the life cycle?
What does this guy look like? Yeah.
Hey,
Dickson, I'll bet it has a complex life cycle.
It has a very complex life cycle. And in fact, it has attracted a wide range of attention from molecular biologists to entomologists to epidemiologists to people interested in chemotherapies. It's a remarkable set of organisms that has actually launched many careers in biology. And so let's begin with a description of the life cycle for all of the Leishmania and then give a general life cycle and then start to give the specifics of how these all differ from each other.
Okay.
All right. So we begin with the sandfly. Okay.
Why is it called a sandfly, Dickson?
Well, Vince, not because it grows in sand. It's not. I don't know why it's called a sandfly, to be honest with you. Although in the Middle East it does breed in places that are sandy and desert-like.
Maybe that's where it was first isolated.
But it doesn't live in the hot of the daytime. Yeah. You know where it lives? It lives at the openings for burrows for animals. So right away, this is going to tell you something. This organism can infect a wide variety of hosts, making it a very versatile parasite. That also means that sandflies, though there must be many different varieties of sandflies, and I'm sure there are, and I'm saying this because I do know that there are, sandflies have specific biting preferences in terms of their hosts to derive blood from, as mosquitoes do as well. But occasionally, the same sandfly species will bite both animals and people, and as a result can transmit disease from reservoir hosts to people. Now, your reservoir host can be as simple as a dog, or it can be as exotic as a gerbil.
So the reservoir host has the sexual cycle?
No, the reservoir host has the same stage of the infection that you do. So that's a reservoir host.
But you called that intermediate hosts for toxo.
You're confusing intermediate with reservoir. So there is a difference between reservoir and intermediate. If you're an intermediate host, that means that part of the life cycle has to occur in you. Whereas if you're a reservoir host,
it's not necessary.
It means that it's reserved over here, and occasionally can jump into you.
So in Toxo, we called the cat the natural host. Was that the name?
It was the definitive
host, and then everyone else was intermediate hosts.
That's correct.
Now here we have the reservoir host.
Yes.
That could be a dog.
Yes.
And what other kinds of host?
Gerbils.
Could be gerbils.
And lots of different other wild animals. If you go into South America, for instance, for their species of cutaneous leishmaniasis, they also have another visceral form, which we will discuss later, called brasiliensis, which is very devastating indeed. But if you look at the reservoir hosts for all of the cutaneous leishmania that are found throughout South America, and you make a list of all of the mammals of South America, it's the same. And interestingly enough, Vince, each one of the species of Leishmania, which can be differentiated now based on DNA biology, attacks a different animal species in South America. So you have one for the armadillo, one for the giant anteater, one for the paca, one for not leopards, but jaguars.
So this is all spread by the sandfly?
It's all spread by the sandfly, but as soon as we encroach into those territories, for whatever reasons, gold mining, settlement making, to make the capital of Brazil, Brasilia, to dig the Amazonian highway, we discover new species by having their sandflies bite us, and we acquire a new form of that Leishmania. And it's been remarkable over the last 30 years, the number of new cutaneous species of Leishmania that have been discovered in South America alone.
Do the Leishmania replicate in the sandfly?
They do.
Oh, they can, huh? Do they harm the sandfly?
It's hard to say, Vince. It's hard. You have to ask a sandfly that question. I'm afraid I don't know the answer. There are some parasites, however, that do harm their hosts, their definitive
vector.
hosts.
So the sandfly is the vector.
The sandfly
is there a definitive host here?
Well, you know, for many years we didn't know whether there was sexual stages in Leishmania or not.
Yeah, are there?
And only very, very, very recently has there been a hint that indeed this parasite, like a lot of other protozoan parasites, has a crypto-sexual cycle that has only recently been discovered through genetic analysis. So the answer is probably. And if it does, of course, then it probably occurs in the sandfly. And that means that the sandfly turns out to be the definitive host,
right.
Like it is for mosquitoes and malaria.
Humans are an accidental host, right?
Well, not so accidental, because the sandfly could pick the infection up from us.
Oh, so we could be reservoirs also.
We can be part of the transmission cycle. Absolutely. You bet. So the sandfly that's infected bites us.
Right.
And as this sandfly bites, it injects an enormous number of pharmacologically active molecules.
I mean, that.
Lots of them. Yeah, from the mosquito biology, they inject vasodilators and blood thinners and anticoagulants and painkillers.
Right.
So you do not feel these tiny little noceums as they burrow and drill their way into your subcutaneous tissues to obtain blood. And as they do so, they inject the infectious stage of this parasite, which is called a promastigote.
Promastigote, which looks to me like a ciliated thingamabobber.
Flagellated.
Flagella, sorry.
It's okay.
It's flagellated.
Yes, it's called. This whole group is called hemoflagellates, by the way.
Hemo? Why hemo?
Well, because they live in your blood.
Okay. So that looks like a single cell.
It is.
With a nucleus.
It does.
And a flagellum at one end.
Correct. And that's what a trypanosome looks like a little bit also.
It's quite beautiful in my view.
We call it a promastigote or a trypomastigote.
Trypomastigote.
Okay, for the trypanosomes you would call it trypomastigode. For this one, we call it a promastigote.
So that's what the fly puts into you.
That's correct, and that's what grows inside the gut tract of the fly and eventually finds its way into the salivary glands. So the point is this, though, that I was raising with regards to the pharmacologically active substances, that either the phlebotomine flies in the old world or lutsamine flies in the new world. Well, phlebotomus is the genus for the old world, and lutsamia is the genus for the new world. But I think they're both phlebotomine in the same family of sandflies. The fly facilitates the biology of the parasite.
How so?
By allowing it entry into the extracellular space by creating this milieu.
No, I see. Just like mosquitoes delivering malaria.
It's a little different, though, because the mosquito delivers the sporozoite directly into the blood.
I see.
This trypomastigote stays, or the amastigote, rather. I'm sorry, the promastigote. The promastigote stays around long enough for the dendritic cell to actually ingest it. It depends on phagocytosis for its life cycle, because unlike the sporozoite stage of the malaria parasite, which is seeking out a hepatic cell, this organism seeks out the phagocytic cell line.
So it stays at the site of the bite, basically, these
organisms,
these promastigotes.
It does. And those pharmacologically active substances are thought in some way to be chemoattractive to these dendritic cells.
Okay.
And in they come and they see something wrong and they start scavenging up all the causative agents, and they get trapped into the biology of Leishmania.
Do the dendritic cells remain there, or do they travel?
Oh, they can travel. Okay, it depends. Now this is largely an effect of the biology of the parasite now, rather than the dendritic cell, because remember I told you there are two kinds, right? There's a cutaneous form and there's a visceral form. Now, in the old world, the visceral form is called Leishmania donovani. In the new world, the visceral form is called Leishmania brasiliensis. And that's where the macrophages, in this case the dendritic cells, migrate away. And look at how specific they are, though. In the new world, the lesions develop along the soft palate and in the urogenital tract. It's remarkable. And it's nowhere else in the body, basically. It might be in a few cells throughout the bone marrow or a few lymph nodes, but mostly it's in these two places. And it erodes away these places, and it gets very ugly very fast, creating all kinds of awful clinical conditions. In the old world form, Leishmania donovani, it goes throughout the entire body and attacks the reticuloendothelial cells of the liver, the Kupffer cells. It goes to the bone marrow and attacks B cells and all kinds of other. So those are the big differences. Okay, the cutaneous forms in both old and new world stay in the skin, or so we thought, until the U.S. troops were stationed in the Middle East during Operation Desert Storm, when Leishmania tropica, which was supposed to remain in the skin, visceralized.
In troops.
In troops.
Wow.
And 600,000 people who participated in that operation were taken off the list for blood donors as the result.
600,000, really?
600,000 plus people who had been in the Middle East and had been exposed, not necessarily infected, were now ineligible to donate blood. And in the Army, that's a big deal because blood is a precious commodity during war. So that was a tragedy for that event.
I didn't know that.
But it also means that whether it visceralizes or not is not just up to the organism, it's also up to the host. You introduce a new host into an old area.
Exactly.
And you get a new result.
Yeah, I was going to ask you whether that was a consequence of naive people who had never seen Leishmania their entire lives moving to an endemic area.
Or perhaps some basic difference in the immunogenetics as to antigen processing or cell migration.
The troops haven't grown up with Leishmania, so...
They have not.
Interesting.
Yeah.
So it visceralized, as we say. And
did.
How many troops got visceral Leishmania, do you know?
I don't think anybody knows the exact answer for that. It was in the thousands, but it wasn't in the hundreds of thousands.
Okay.
But it was enough to cause concern in the Army as to, my gosh, we have to do something about this now, too. Not only do we have to worry about malaria, but now we have to worry about... And again, guess where we are again? We're right in the heart of that whole area.
Sure.
So we're in Iran and Iraq. And
we don't have any...
Iran,
do we have drugs to treat Leishmania?
We do, yeah. We'll get to that, too.
So,
okay, the organism now finally gains entrance into the subcutaneous tissues, into the extracellular space. The dendritic cells take them up. And then two things can happen. One is the migration of dendritic cells away from the area of infection, or the dendritic cell stays put, and the organism now goes inside the parasitophorous vacuole, and something interesting happens then, Vince. The parasitophorous vacuole starts to acidify, and the lysosomal enzymes start to fuse. But the parasite contains stronger lytic enzymes and destroys the enzymes that are produced by by the lysosomes.
Yeah.
So its escape mechanism is that it's just got bigger and better, robust acid hydrolases, and it hydrolyzes the hydrolases.
Wow.
And it stays put.
And those more powerful hydrolases do not touch the Leishmania, of course.
Oh, but of course not. They're totally protected against all this stuff, right? So here they sit in what should be a digestion vacuole, and they start to reproduce. And little is known about its metabolism. Very little is known about its metabolism.
What does reproduction constitute?
Well, we can't. Well, binary fission.
That's it. Asexual, right?
That's right. It's not like malaria. Malaria is its other weird kind of division cycle where all the nuclei divide at once in the same cytoplasm, and then this waste substance called hemazoan is produced.
Right.
In Leishmania's case, there is no waste production of hemazoan because they don't feed on hemoglobin. So what do they eat?
What do they eat?
Well, Vince, I wish I knew the answer to that. And I wish everybody else did too, because
clearly something from the cell.
Might be able to interfere with it, but it's got to be something from the cell that the cell donates to the parasite to make it grow bigger. But it's not the actual budding off of cytoplasm. This thing doesn't ingest whole pieces of dendritic cytoplasm and eat it. So we don't know how it feeds, basically.
So it just divides, and what does it do, make more promastigotes?
No. In this case, the moment it gets taken up by a dendritic cell, the flagellum disappears.
It doesn't need it anymore, does it?
Nope. So the term mastigote
Yeah.
relates to the flagellum. So Mastigophora is the phylum of protozoa that are flagellated.
Really?
Yeah. So without a flagellum, what would you call them?
Aflagella? Amastigote.
Bingo.
Sorry, why did I say
No, Vince, that's okay. No, you're actually right.
Yeah.
It's the Latin word for a flagella, so it's amastigote. So from now on, these organisms are missing the flagellum. They've got a remnant, all right?
Interesting.
They've got a remnant, but they don't have the whole apparatus.
So inside the parasitophorous vacuole, the flagellum is lost.
Correct.
Becomes an amastigote, and then as they divide, do they not have flagella?
They do not.
How interesting that they've turned off. Do they look morphologically different from promastigotes?
Oh, they're totally different. They're rounded cells with a little kinetoplast. It's called a kinetoplast.
What's that?
It's a collection of maxi and mini circles of DNA.
DNA.
And mini and maxi circles have quite specific functions.
And these are separate from the nuclear DNA?
They're quite separate.
Well, are they like mitochondrial DNA?
It's derivative of, but not necessarily the same as.
The name implies motion.
That's right. Kineto means plast. Kinetoplast, a motion plast, or something like that.
What does the plast part mean, Dickson?
Oh, it's just part of the plasm, I think.
Okay.
So people have made careers out of studying the kinetoplast and what it does. People like Larry Simpson and Ken Stewart and other people. And in fact, that's where we learned about RNA editing, from studying what these mini and maxi circles actually do and their transcription factors and all kinds of interesting molecular biology.
RNA editing, of course, is the change in a sequence that is not templated.
That is correct.
So you make an RNA, and an enzyme comes and changes one of the bases later.
Correct.
It's called RNA editing. It happens in viruses, too.
Exactly.
It's called a post-transcriptional modification.
That's incredible.
Why is that incredible?
Well, because the genes are supposed to tell you what to do, and here we are disobeying.
Yeah, it's amazing.
And it's a way to have the last word.
In the Leishmanias, what is the consequence of this editing? Do we know?
We wish we could grow this thing in cell-free media so that we could study all of its metabolism.
Can't do it, eh?
We can grow the promastigotes very nicely,
I see.
Extracellularly, because they are extracellular parasites in their definitive host, the sandfly. But these are intracellular amastigotes in the dendritic cells. And I don't think anybody's learned how to grow these things cell-free yet.
So this cycle so far could happen in a human, a horse, a dog, a gerbil. Is that what that is in the picture here?
It is.
Any number of hosts.
That's right. Or a jerd. There are jerds out there as well.
And the consequence is a skin lesion.
For some of these species, and for others, they go visceral.
So they have this young lady here with a skin
Yeah,
lesion.
she's got an unfortunate scar.
Big one on her face, and they stay. The scar remains,
does
right?
not go away.
What causes the lesion? So it must be partly an immune response, right?
Well, it is, and it's a hypersensitivity response that's generated against the byproducts of the organism, and it results in an increase in the number of epithelial cells, which then become fibrotic and stay put. They just they stay as a fibrous lesion. So the life cycle continues, though, Vince, because the organism somehow has to get its way into another sandfly in order to be retransmitted. Okay, so some of these dendritic cells in the visceral Leishmaniasis form, because I must tell you what happens clinically too, because that helps describe where these organisms end up. When the lesion first develops in the skin, they're indistinguishable. Visceral versus cutaneous are absolutely indistinguishable.
So even in a visceral infection, you will have a cutaneous stage.
You begin with a cutaneous stage.
Okay.
That's correct. And it starts to look like a craterform lesion, then it just disappears. And they say,
I'm fine.
Must have been something else. It's tragic, because when that goes away, that means it's someplace else.
So if you're just having a cutaneous infection, you will have scars, you're saying?
You bet. If you don't treat them, you will.
And then if you don't have scarring, it's likely going to a visceral form.
That's right. Now, it raises the big question, though. How does the infection get retransmitted? I mean, how do you take a visceral Leishmaniasis case and get that organism into another uninfected sandfly?
Must be a blood phase, no?
Right.
Sandflies take blood meals?
They do.
Okay.
That's what they do to make eggs.
Yeah.
All hemophagous organisms use their blood meals primarily, some as food also, like black flies, but they also use it for egg production. So here we have the sandfly now needing to get an infected cell with the amastigotes so they can transform into some other stage inside the sandfly, right? The cutaneous form is easy to see because as this lesion spreads and gets bigger and bigger, the sandfly can just feed at the living edge of this and pick up the dendritic cells that are harboring the organisms and fly away and start the infection again. But the visceral form is a little more cryptic. And so what it means is that you've got to have circulating cells at all times with the organisms inside them for the organisms to be taken up by the sandflies as they obtain their blood meals. Complicated stuff, but nonetheless, it's a very successful infection indeed in both cases.
So both cutaneous and visceral forms have circulating amastigotes?
No, not the cutaneous.
Just so that can't be transmitted.
It can, only by the sandfly feeding at the living edge of the lesion.
Okay.
Right.
Did you say that?
I did.
My apologies.
That's okay, Vince. It's okay. It's good to reinforce this by repetition.
And in the visceral form, you have blood.
Correct.
Amastigotes, it would be, or promastigotes?
Always amastigotes.
After the infection.
Yes.
So then the sandfly picks up an amastigote.
That's correct. And then it undergoes transformation into the promastigote stage.
Somewhere in the gut of the sandfly.
That's correct.
Why does it need to be a promastigote? Seems to me it doesn't. It could just be carried by fluids.
I think the answer, we have no answers for why questions in biology, Vince.
We don't have any answers?
Not even one, like, why is life?
Oh, that sort of thing.
That sort of thing, yeah.
Well, I can answer why do viruses infect us.
Yeah, well, I think the reason why it develops into a promastigote stage with a flagellum is because it actually swims in the gut tract of the fly. Now, interestingly enough, that there are several different stages of what you would call a promastigote, but they're given different names. In trypanosomes, they have a much more definable set of stages, like the metacyclic trypanosome and the. And I'll relearn this, of course, by the time we're on to the next episode, but because it wasn't my specialty. My specialty was Trichinella. Everybody out there knows that. So, but I'm doing okay so far with Leishmania. I hope I don't affect all those Leishmaniacs out there in an adverse way. I am featuring your organism right now, but if you have something to add to it, of course, you know how to get in touch with us. The way this organism reproduces is by attaching to certain numbers of gut cells at a certain level of the gut tract. And the sandfly has 3 levels of the gut tract, a foregut, a midgut, and a hindgut. For some organisms, that are trypomastoid stages, they actually are defecated out of the organism in order to infect. And Trypanosoma cruzi is one of those organisms. And they will migrate down the gut tract of the reduvid bug and transform into the next stage by attaching to a different lectin as they change their surface proteins, which is quite amazing. And I have a feeling that Leishmania do the same thing in sandflies. And I think there are people out there that are studying the intimate biology of the growth of this parasite within the sandfly itself. And by learning more about that, maybe we can learn ways of interfering with the biology at the sandfly level. And, feeding something to dogs, for instance, that would interfere with the ability for them to transmit the infection.
Is this a common dog parasite infection?
It's extremely common.
So that's what Sophie was talking about. Her
right.
Spanish dog had severe
If you draw a ring
skin parasites
all the way around the Mediterranean basin, you will find this is an endemic infection all the way around.
But in a dog, you might not see it because of the fur or the hair, right?
Yeah, that's right.
Is it?
Although you'll find it around the face mostly of these animals, because that's where the sandflies find naked skin.
Yeah.
So you will see lesions on the nose and around the eyes.
Do you want to treat it?
Well,
in a dog?
You could.
Do you have to, or is it going to be fatal?
Well, I think no, it won't be fatal. There are good drugs for this, and one of them is sodium stibogluconate. That's the gold. I was going to think of the gold standard, but it contains tin. It's the tin standard
for dogs or humans or both.
Both. For both.
So will it become visceral in a dog?
That's a good question, and I think there is a variety of Leishmania called Leishmania infantum, which is found around the Mediterranean basin also. And yes, that is a visceralizing form of cutaneous Leishmaniasis. And that is the form, I believe, that got into the dogs that are now in this world. So let's go to the PubMed and we can actually...
If I go to Greece, should I worry about getting Leishmania?
I wouldn't worry about it, but I'd be a little bit concerned.
If a sandfly bites me, what should I do?
Well, we're going to get to that later. It depends on
if I came back here and I suddenly developed a lesion, should I go to the emergency room?
No. Go to the dermatology clinic, and they'll take a scraping from it, and they'll culture it in Schneider's medium, which is an insect tissue culture medium. And if you've got it, the promastogote stage will grow up in that, and then they can make the diagnosis and give you sodium stibogluconate.
All right. What did you want me to look up on PubMed?
Look up introduction of Leishmaniasis into the United States via hunting dogs.
That's a lot of search terms, Dickson.
Okay, make hunting dog Leishmaniasis. And you'll get it. It was first introduced into Kentucky, as I recall, and then the epidemic spread, and now it's an endemic.
Yeah, now we got it.
It's an endemic infection.
You wanted the U.S.?
Yeah.
Because I got a lot of articles on Spain and Greece.
Of course.
Different dog populations in those countries. In fact, is Leishmania become endemic in the U.S.?
The answer is yes.
Let's see. But this is an old article. It's 10 years old.
Right. Well, that you can find a newer article than that, that's for sure.
Do you think?
I do.
Here we go. Believed to be all but absent from the U.S.,
right.
Leishmania parasite has infected more than 1,000 hunting dogs in 21 U.S. states in the Canadian province of Ontario.
Bingo.
Nobody is claiming that the disease is about to run riot in the U.S., but the widespread outbreak in dogs has experts wondering whether visceral leishmaniasis, which sickens over 500,000 people yearly in South America, Africa, Mediterranean, and India, has become an endemic disease in North America.
Right.
In dogs.
Does it list the species there?
The species of dog?
No, the species of...
I'm sorry.
It's Canis domesticus. I know which species that is. There's only one species of dog, Vince. Did you know that? There's only one species of dog.
Why is that?
Ask Charles Darwin.
Well, Chuck is dead. I can't.
I know. Well, okay. Ask his progenitors then. Dogs are totally free to interbreed with each other, and they were all derived from wolves to begin with, we think, and they all share a common genome.
Well, this is fascinating. Leishmania infantum.
That's the one.
This is late 1999.
In Kentucky.
Foxhounds at an exclusive hunt club in Millbrook, 130 kilometers north of New York City, came down with symptoms such as bleeding, wasting, seizures, hair loss, and kidney failure. 24 died, and it turned out to be Leishmania.
Right. I also heard about the introduction in Kentucky, too. So there's been a number of introductions, actually, and it just has to be lucky.
How did it get to them in the first place?
Well, they bought them from European sources, and it's just like West Nile virus got here.
Sandflies.
That's right.
Interesting.
They were sitting here waiting for the introduction of that parasite. And bingo, here it is.
So this was the only article I found using the search terms.
Well, try Leishmania infantum in the USA.
Hunting dog Leishmania. Maybe I should just put Leishmania, dog Leishmania US. How's
Sure,
that?
you can do it that way too.
Now we have 11 articles.
See, we don't have to know all of this material in order to know the answer, because we know where to go for the answer. And that's why the internet is such a powerful tool.
This is a 2009 article.
That's
New means
relevant.
of canine leishmaniasis transmission.
Uh-huh.
In North America, the possibility of transmission to humans still unknown.
Right. It's rare in Europe even, and it's very rare for people to catch it.
I will make a prediction.
Please.
It will,
of course,
go to people in the U.S.
You bet.
You would agree.
I would. And by the way, you can transmit this by transfusion also. So.
Do we test blood for this?
Hell no.
Why not?
Oh, I'm sorry, audience. Heck no.
Because it's not a big deal in the U.S., right?
Oh, that's right. I mean, it's not enough. There's not enough there to test. And how would you test for it?
Well, you could develop an assay. That's not a problem.
Well, there is tons of them. Even a PCR assay that would be good, but it's very expensive. Best way to get blood transfusions are to donate your own blood and then ask for it later. A lot of people do that for pre-op surgeries now. They're voluntary.
Sure.
At any rate, back to the life cycle.
Okay.
Okay, so somehow we have to get a cell that's infected with Leishmania into a sandfly. In the cutaneous form, it's from the lesion that forms at the skin level. And for Leishmania brasiliensis and Leishmania donovani, it's from the circulating, not dendritic cells anymore, but other macrophages like neutrophils even. This will infect any phagocytic cell. So that's a powerful biology for this organism. But you asked me whether or not this was more popular than toxoplasma. This infects reticuloendothelial cells. Toxoplasma infects every cell.
Every cell, yeah.
Including reticuloendothelial cells, but everything else too.
Right.
So it's a versatile parasite that causes horrible disfiguring diseases or life-threatening diseases.
So disfiguring because you have these lesions on your skin.
Yeah, well, if you talk about
this fellow, he's lost his soft
He's lost all of his soft palate. That's correct. And that what you're seeing here is a picture of a poor little kid from rural South America who's had the visceralizing form of that called Leishmania brasiliensis, and the infection is old. That means that this little kid hasn't seen a doctor in all of his life. And suddenly somebody says, oh, well, there's a doctor that's coming upriver, and you need to go see them. Believe it or not, they can treat this patient and do reconstructive surgery, and you can almost return to a normal face. So it's quite dramatic. This is a dramatic infection. In India, by the way, where this infection was first described by Leishman and Donovan, it's characterized by an enlarged spleen and an enlarged liver. You get hepatosplenomegaly with this one. And that's as opposed to malaria, which only gives you a large spleen, or schistosomiasis, which only gives you a large liver. So you can now...
Differential.
You can make a differential just based on physical findings sometimes.
Can you transmit the infection by contact?
Good question.
So are the lesions infectious?
Not likely. It's not like ringworm, or it's not like some of the other fungal infections where, like Tinea pedis or Tinea capita or something like this. It's not a dermatophyte like that.
Because I ask because I see for brasiliensis there are urogenital lesions.
That's true. So would it be a sexually transmitted infection? I think someone with this infection in that area would not feel like having sex, to be honest with you. This is a painful, nasty process.
All right.
In that area. Now, in the nasal zone, by the way, that's totally anergic. There's no sensation of this. That little kid doesn't feel any pain whatsoever.
He's missing his nose and mouth.
Exactly.
How could he not feel pain?
Exactly.
Has the parasite done something to...
We don't know what the pathology is all about, but
Wow.
we do know that there's these lesions are painless.
All right.
So it's remarkable. Okay, so that raises the question of how do you make the diagnosis? Okay, let's say you're a clinician, and you want to know whether your patient, you raise this by saying, I come back from Greece, I've got this lesion on my hand, what do I do? Okay?
You told me to go to a dermatologist, right?
Go right to a dermatologist.
Actually, I would first ask you, I'd say, Dick, should I go get this checked?
And I'd say absolutely. If you can ask the question, you already know the answer. It doesn't go away. It's getting bigger. I think I should go see somebody. Now, I must tell you that we had a technician in our laboratory. Her name was Susie Azan. She was a remarkably wonderful person and just loved working with parasites and cell cultures and everything else. And we had two people in our group, Suzanne Holmes Giannini and Dr. Philip D'Alessandro, who was my chairman, who worked on Leishmaniasis. And Susie Azan was in charge of the animal transfer of the infection. And she worked with hamsters, because hamsters are susceptible to this too, and you can induce dermal lesions with it. And they were working with a cutaneous form of Leishmaniasis, and she accidentally stuck herself.
Mm-hmm. With a promastigote prep?
That's right. She knew that she had, just like a phlebotomist fly or Leishmania fly would bite you and inject them, there she had it right in her middle finger. And she knew this wasn't going to go anyplace. It was just going to sit there. So what we did was, when Susie stuck herself, she knew that she would get a lesion. So we all sat around and discussed whether or not she should let it grow up or not. And she said, "Okay, let's watch it. Let's watch it grow." So, she was game, and this could have been a lot worse if it was Leishmania donovani. We would have panicked right there and said, "Go take some sodium stibogluconate now." But the fact is that the lesion did develop on her finger, and she was fascinated by this. That's the sign of a good biologist, by the way. Somebody who's not afraid of their own parasites, but I'm sure if you work with HIV, you're just the opposite. So the point of this story was that she eventually ended up wanting to get rid of it. Now what do you do?
So she had a scar. Not yet.
Not yet. She had a developing lesion, and the lesion was getting bigger. Okay, it's big enough now. I've seen it. Ha. Yeah. We can isolate organisms from it. That's what it is. That's fulfilling, not quite co-postulates, but almost. Yeah. So now you have to get rid of this thing. So there are some benign ways of trying to get rid of this. The first one is to submerge your finger in a very hot glass of water, like 105.
And then you drink the water afterwards?
No, Vince. You just depend on the heat killing the organisms. And that was tried, and the lesion did stop growing for a couple of days, but then it started to grow again. So then there are a number of drugs that you can try for this, okay? And the first drug, of course, is sodium stibogluconate.
Oral?
It's a heavy metal-containing drug, and it's toxic, not just for the organism. It's toxic for you, too. But there's a huge therapeutic difference between the amount needed to kill the organism and the amount needed to kill you. Nonetheless, there's some toxic side effects, and she didn't want to do that, so we actually ended up giving her ketoconazole, which is an antifungal agent, strangely enough, and it's very effective against this parasite. And the lesion went away, it resolved, and she has now a tiny little scar on her middle finger. That's proof of the fact that she was infected with cutaneous leishmaniasis.
How long did it take from the stick to the first sign of a lesion?
About a week.
Okay.
And then after that, the lesion just kept getting bigger.
And this is over the course of weeks?
About a month. She let it go for about a month, and then she cured herself.
So now she has antibodies to Leishmania antigens, I presume?
Well, it's been quite a bit of number of years now. It's been about 25 years since
so
that
the question is, once you get it, can you be reinfected?
No. This is the beauty of this particular infection. The answer is you cannot, and we're not exactly sure about the mechanism of protection, but it's lifelong. And that's why scarification works.
Right.
And that's why in some Middle Eastern countries, when somebody has a resolving lesion, they take that person and they inoculate all of the young women in places that the lesion would never be seen, not your face, like the buttock or the behind the heel on the foot or some other place like this, and allow that lesion to develop and resolve. And that means from that point on, you'll never get another lesion. Now, it's too bad that doesn't work against the visceral form of this, but it doesn't.
I forgot to ask you that question for toxo. If you get infected, do you get it again?
The common thought among clinicians and immunologists is that, yes, you can catch it again.
And malaria?
Oh, many times. Everybody can catch that.
Of course, they get it over and over.
Immunity wanes within weeks after the
But not Leishmania.
resolves.
Now, let's say you're infected with Leishmania donovani. Can you get infected with Braziliensis? Yes. So if you go cross-species, there's no protection.
None. You can also get the cutaneous form, too.
If you get the visceral form,
I believe, although I'm not certain,
it can be fatal, right?
Oh, it's about Well, untreated, it's in the neighborhood of 30 to 50%.
Okay, so those who recover then are going to be immune to subsequent
right.
cutaneous or...
No, just to the visceral.
I see.
There's no cross-species protection.
Got it. Now here in this picture of Leishmania donovani,
yes,
this fellow at the bottom here has a funny thing on his face. Should we talk about that? Or?
Sure, we shall.
Or do you want to wait?
No. This has many clinical manifestations. Depends on your immune system. Mm-hmm. So if you have a normal intact immune system and it's very active and very directed, okay, and it recognizes all the antigens that it should, then this will not be a fatal infection. You will have suffered, but everything will go away and you'll be normal again.
I see.
On the other hand, if you've got some immune defect, then you can develop an infection which has similarities to leprosy, which also, I think, has an immunologic component to it in terms of immunogenetics. So that people with a certain haplotype and a certain range of MHC will develop a form of tuberculosis called lepromatous.
Right.
Okay, and that expresses itself as what they call lion face, right? Because it localizes it into the skin, and when you isolate it from these lesions, you can find huge numbers of the leprosy bacillus. The same thing happens to Leishmania if you partially treat in these individuals. If you give them just a little amount of drug and kill off most of the organisms, it sort of drives it into the skin. And you get this post-Kala-Azar dermal leishmanoid reaction to it. People out there think I'm a clinician when I talk like this.
Kala-Azar. What did that come from?
Kala-Azar is, I believe, a Hindi word for black fever. Okay, so it does it in many respects like plague that creates these buboes, and then they necrose, and you get these large dark lesions under the armpits. This organism also gives you this similar result in lymph nodes, and you get these dark lesions, and you have a fever.
Beautiful.
So there's the word Kala-Azar. It's also called dum-dum fever because there's a town in India called Dum-Dum, and it was isolated first from that town. So they have a number of these little words to go along with them, but the post-Kala-Azar dermal leishmanoid reaction in this individual is totally curable by, guess what?
Immunosuppression?
No, there's this wonderful weird stuff that's never been fully characterized called transfer factor. Remember that?
I do remember it.
Transfer factor was thought to be a combination of an RNA extract from immunologically competent cells and a little clip of the antigen itself. And I can remember some of the people who worked on transfer factor. And if you make an extract of lymphocytes and isolate what you would call the transfer factor and give it to these people with post-Kala-Azar dermal leishmanoid, it clears right up. They get better. Or if you give them more drug.
Yeah.
And then it clears up. Whereas with lion face, Leishmania, or with leprosy, Hansen's disease, as they call it, it doesn't clear up.
Now, I don't know if I think I interrupted you. You had originally started with, if you suspect you're infected, what should you do?
Oh, you want to know about diagnosis? Sure, okay. So
I don't think we finished that, right?
We did not. So the cutaneous form is pretty straightforward. If you come in with a lesion that continues to grow and you've been in an area that's endemic for cutaneous leishmaniasis, and that's a lot of the world, as you can tell now, then your best bet is to go to a good clinical dermatology laboratory that's connected to a large urban center, because they're used to seeing these from travelers and from immigrants from those regions, and also from people that go there on their vacations. So that's one approach. The other, of course, is that if you develop these fevers, and they're cyclical fevers, by the way.
Oh, I meant to ask you, if you get a fever.
You get a fever.
Do you get it at the...
Kala-Azar, that's black fever.
So, at the cutaneous form?
No, you don't get a fever.
Just visceral.
Just the visceral. But the visceral is, of course, much more serious. So how would you know that you had visceral leishmaniasis just based on a fever?
Right.
But these fevers are periodic fevers. These are like very, very regular fevers. And as a result of that, they're called dromedary fevers.
Dromedary.
Okay? Yeah, they're...
like a camel.
That's correct.
Up and down.
That's right. That's exactly right.
Interesting.
Yeah. So it's got all of this... I guess you'd call it medical folklore associated with it, but the fact is that if you've been to an endemic area and you've got a fever and they did your blood smears and they didn't find any malaria, which they suspect to begin with,
Right.
then they have to think about something different. Could have been relapsing fever, right?
Which is
caused by a spirochete, treponema.
Yep.
Or it could be a viral infection, right? A lot of viruses induce fevers.
Sure.
But it could also be visceral leishmaniasis. So how would you actually test for visceral leishmaniasis?
I'd take blood.
You wouldn't find it.
No.
Nope. Sorry. Take bone marrow.
Wow.
Bone
That's
marrow.
a big deal, Dickson.
No kidding. But it's a big deal when you haven't found out the cause of this fever and you're getting sick and you've got hepatosplenomegaly and you've got lots of things going on.
So you do a bone marrow.
They take bone marrow, they make a smear,
Wow.
they do a stain with Giemsen, they look for the organisms, they look for the amastigotes, but also they take bone marrow and they inoculate Schneider's medium, which is an insect tissue culture medium, and they incubate it at room temperature because insects live at room temperature. And in about 3 days to 5 days, the amastigotes will transform once the cells disintegrate around them.
You got cells in this culture, right?
Oh, yeah, you have to have
Insect
cells.
Yeah.
Not insect cells, no. You've taken bone marrow.
Oh, there's no insect cells in the insect cell medium.
None, but it imitates the environment of an insect.
I see. Okay.
So it fools the amastigotes into thinking they're in the midgut of a sandfly.
Right.
And they transform into their promastigotes and start to reproduce. You can get a test tube full in about a week.
Wow, it's really bone marrow. Eh?
Yeah, they do blood too. I mean, you'll take some blood and do this as well. Or you can do a PCR.
On blood?
Yeah. Or on bone marrow.
Yeah, but there must be some organism in the blood, right? As you said, that's how the sandflies pick it up.
Correct.
It's a timing thing.
That's right. So PCR is a very sensitive test, and it's a very specific test.
Yeah.
And they're using kinetoplast-specific probes so that they're sure of not picking up DNA against lots of other things that might be in your blood.
Wow.
So I think the whole system is interesting and it's fascinating, especially with regards to the way our immune system handles this infection, because there are lots of people that make their living studying the interleukin responses that are elicited by this infection from time zero till the time an animal is cured or until it dies. And they have lots of different strains of mice to work with, and the fact that this will infect mice quite nicely because it's pan-host specific, so that it's got a very, very wide range of hosts.
Is this a problem in people with AIDS?
Yes. Yes, it is.
I would guess so.
It is.
How do you know so much about it if you didn't teach it and you never worked on it?
Well, Vince, it's called guilt by association. You know, if you're the only worm person and you're sitting in the presence of 4 protozoologists that all work on it and that's all they talk about, you can't help but learn something.
I see. You absorbed it over the years.
Sure, and plus I wrote the entire protozoan section to the textbook, and the reason why I did that is because I wanted to learn the subject. So
Beautiful.
I have a lot of good friends. Dennis Dwyer is one of those, and I hope he's listening to this, and he's a big expert on leishmaniasis that works at the National Institutes of Health. He's the lab of cell biology section head for the NIH Laboratory for Parasitology. So he and I talk regularly, and I use him as my fact checker, so to speak, and he's abreast of the situation. And there are lots of other people, and I also had the privilege of running a series of Gordon conferences with Elmer Pfefferkorn from Dartmouth, rather. And half of those talks were on protozoan infections, and most of them either featured trypanosomes or Leishmania. There were a few on toxo also, but most of the people thought they were obliged to put their intellect where their mouth was and work on something that had relevance for humanity. And Leishmania is one of those subjects that we still haven't licked it, so we're still working hard to try to get rid of it.
Excellent. Anything else, Dickson?
There's tons of more stuff to talk about with this parasite because I've just scratched the surface, no pun intended.
So we can do another episode?
We could. We could actually have a guest.
What would we talk about?
Well, I think we should talk about the mechanisms of immunity to this thing, and I think for that one we
need a guest?
We'd probably need a guest to come on and talk to us about that one.
Well, let's do a few emails before we say goodbye.
Okay, and do our pick of the week, too.
Sarah writes, to my favorite scientists.
I like it.
Does she mean us?
I don't know. Is there anybody
or
else
to
here?
everyone listening. I am a high school student from Serbia, and microbiology is my passion. I plan on going to university and studying it. I wanted to tell you both how much I enjoy the TWiP podcast.
Great.
I especially enjoyed the one about tapeworms. I urge you to do the show weekly. I can't wait until the next one. Keep up the great work. Thank you, Sarah.
Sarah.
Next one's from Jeffrey, and actually is incredibly relevant to this episode.
Well, then, read on.
Jeffrey writes, you may have already been alerted to this one. It's a paper in Insect Molecular Biology.
I love it.
Great journal.
I love it.
It's entitled Flying Vaccinator. A transgenic mosquito delivers a Leishmania vaccine via blood feeding.
Whoa!
You hear about this?
It's not even a vector. That's not even the vector.
In Japan, they used engineered mosquitoes. So they made a transgenic anopheline mosquito that expresses a Leishmania protein, SP15,
cool,
in its salivary glands. Mice bitten by these mosquitoes make antibodies against the protein, but they haven't done any challenge experiments here. Let's take a look very quickly to see if they've done challenge. I think they've just looked at the induction of antibodies.
Right.
Here, let's see, full text, HTML.
But SP15 sounds like it might be a protection-inducing antigen.
SP. I'm not familiar with this. Generation of transgenic line, expression, location of the protein, antibody responses in mice. End of paper. No protection. But I guess this
That'll be their second paper.
Antigen is involved in protection. So it's a proof of concept.
Right.
Now, Dickson.
Yes, Vince.
Let's say this mosquito induces protective antibodies.
Okay.
Would it be permitted to release it in the wild?
Good question, because anopheles carries lots of other things.
I can't imagine that this would happen, but what do I know?
It depends on how. It depends on. Yeah, it would depend. I think if you could get that mosquito to also not transmit malaria by genetically manipulating it in another way, then you could have the ideal transfer of protection.
Actually, a paper has come out showing how to modify a mosquito to make it resistant to malaria.
Right.
So if you could use that combined with this.
Bingo. But they could also still transmit filariasis, which is a worm.
Yeah.
So, you still have to worry about some things. And, of course, there's a lot of viruses that can transmit also. Mosquitoes are flying syringe needles, and it's really hard to imagine how their swords could be turned into plowshares. But here's one attempt.
Oh, what a nice phrase. Swords to plowshares.
That's an old phrase.
Yeah, no Dickson, but I like to hear that.
Thank you.
The next one's from Prasad, who writes, Hi, Dick and Vincent. I love your podcasts, TWiP and TWiV, although I am a virologist and think that viruses are the most amazing things. With your podcasts, I have come to appreciate the parasites as well.
Great.
I think Dick will like to know that I work on dengue.
I would love to know that.
My recent PubMed search led me to this article. In Current Biology, titled Plasmodium falciparum accompanied the human expansion out of Africa. It talks about the origins of P. falciparum. I know that TWiP has moved on to other parasites, but it would be great if you can explain the controversy of malarial origin a little bit. In any case, I'm over in Singapore currently, and if any of you are in this part of the world, drop me a mail. I can show you around.
Ha!
Cool. Thanks for the podcast. Keep them coming. I've never been to Singapore, have you?
Nor I, but there's a lot of interest in Singapore for the vertical farm project, so maybe I'll get there.
Oh, that'd be great.
Yep.
So, Dickson, what do you know about this expansion of P out of Africa?
Actually, there's a paper that just recently came out that said that we acquired Plasmodium falciparum some 2.5 million years ago as the hominid lines split off from the ape-like lines.
Mm-hmm.
But what relevance that has for making it out of Africa, that's hard to say.
Let me read you this abstract.
Please.
Here we analyze a worldwide sample of 519 P. falciparum isolates sequenced for 2 genes. So they want to try and track the phylogeny of these organisms by looking. We find a strong negative correlation between within population genetic diversity and geographic distance from sub-Saharan Africa. In contrast, regional variation in transmission intensity seems to have had a negligible impact on the distribution of genetic diversity. Age estimates for the expansion of P. falciparum further support that anatomically modern humans were infected prior to their exit out of Africa and carried the parasite along during their colonization of the world.
That would make sense.
I don't know why there is any controversy about that. Do you know?
Nope.
Have you heard of it?
Nope.
Would you have heard of it?
Not really. No, I'm not following the literature as closely as you'd need to in order to hear about it, I'm afraid.
Let's see if we can glean anything else from this wonderful paper here. And I'm not being...
No, I think any light that you can shed on this dark subject would be good.
I see the age has been highly disputed, with previous estimates for the most common ancestor spanning more than an order of magnitude, with values ranging from 10,000 to over 300,000 years. So it's an exclusively human parasite with no known animal reservoir, P. falciparum. Are they right?
That is correct.
We hypothesized that if it had been associated with humans for over 50 to 60,000 years, its current structure could still carry a signal of human settlement history. And that's what they've looked at here by looking at all these isolates.
Yeah, well, humans, as we know us, have only been a human species for about 175,000 years, something like that.
Seems to me that this is quite compelling, and that makes perfect sense that it would have come out with population. Humans began in Africa, right?
That's true.
And they migrated outwards.
Yep.
They bring their parasites with them.
You bet. That's what I predicted killed off the Neanderthals. Not malaria, but other similar infections that we brought with us.
Dickson, I'm reading a book called The Red Queen. And it was my pick of the week last week in TWiP. And it posits that the reason sexual reproduction evolved was to protect against parasitic infections. And there's a lot of evidence that that's the main reason.
Interesting.
Because asexual reproduction produces clones which are all susceptible to the parasite. And the only way you can get resistance is by mixing.
Recombination. Yeah.
Spencer, who is a physician in New Jersey, writes, I often see patients with addiction problems. Now, 1 month into TWiP withdrawal, I can sympathize. Please press onward with this fascinating podcast series. I am a cholesterol and lipid specialist and run a testing and referral center in New Jersey where I see patients with complex lipid and metabolic disorders. Are you aware of any crossover from the microbial world in terms of inflammatory influences on the vasculature that can lead to advancing atherosclerosis? For example, we know that an inflammatory marker, C-reactive protein, which has been precipitated from patients with pneumococcal infection, may be an independent marker for atherosclerotic risk and additionally may be a target of therapy.
True.
Do you know anything in the parasitic world, Dickson?
Well, chlamydia are thought to be targets for that kind of therapy too, because they induce inflammation of the vasculature as well, and using things like aspirin, which would keep down the inflammation, would allow the infection without inflammatory response consequences. There are lots of parasites that affect the level of lipids in your blood system, however. And I mentioned filariasis before. We haven't even talked about filariasis.
Not yet.
But we will. And when we do, we'll talk about one that induces lymphedema to the extent of the accumulation of huge amounts of lymph behind the blockage. Right? And as a result of that, patients will develop lots of lipids in their blood, which they shouldn't have because they accumulate and then they just spurt out, so to speak, from these large lesions. But yeah, that's
well. There are a number of cases where viruses are thought to be involved in inflammatory diseases.
Sure.
Atherosclerosis, particularly, there's one virus, norovirus, in mice, which is thought to be involved in obesity and insulin resistance.
It's probably from Norwalk, Ohio, isn't it, Vince?
Atherosclerosis as well, and this is being studied in the mice.
Right.
So, yes.
The one I referred to was Wuchereria bancrofti, by the way. Named after, again, two people, Wucher and Bancroft.
Thanks for that email, Spencer. And here's one from Mitch.
Yeah, we're sorry to dole all of these TWiPs out in less than satisfactory levels, but we'll try to do better in our
old age.
In our old age.
Mitch writes, I came across this joke online today and thought you guys would appreciate it.
Uh-oh.
Taenia Solium walks into a bar and the bartender says, "We don't serve your kind in here." Taenia Solium replies, "Well, you're not a very good host." Mitch. Thanks, Mitch. You're not a very good host. Taenia. Tapeworm.
And yeah, I know.
You don't like that one. He's frowning.
Actually, I don't. The one I like, I like a lot of them, but I didn't like that one particularly, but nonetheless. Forget that. Actually, one of our colleagues produced an X-rated parasitology textbook.
Really?
Which I can't.
Never heard about
I can't tell you any of those jokes, but they were all very clever.
Kieran writes, I just wanted to say thank you for all your time and effort put into the creation of TWiP and TWiV. They represent the very best in podcasting and show exactly how hugely beneficial they can be. It's great. Thank you. TWiP is especially fascinating and illuminating, and every 2 weeks is not enough to do justice.
Everybody says the same thing.
To the breadth and interest of the subject. It would be brilliant here to hear something of fungal parasites, especially with the wide range of strange and distinct behavior they exhibit, e.g. Cordyceps unilateralis, and the effect it has on its host's behavior.
Well, we'll have to get a fungologist in here.
I think we should do TWiF.
TWiF.
This Week in Fungi. That's right. I would love to do that. Get a fungal
It's actually
expert.
TWIM.
Mycology.
That's right, Vince.
Well, we could do either one.
TWIM.
Yeah, mycology.
TWIM.
I could do one on, we do TWiV, TWiP, TWIM, and TWiB, bacteria, and then a fifth one, I don't know. We'll see.
You have to do medically important arthropods.
This Week in Insects. Thank you again for the great work, and yours very humbly, Kieran. Thank you, Kieran. And the last one is from Mark.
Okay.
Thank you for a wonderful podcast series so far. By the way, Dickson, you see, people love this.
Indeed.
I know you're a busy guy.
It's hard to believe, isn't it?
But whenever we can do it, Dickson.
I think
actually, once you've built a few vertical farms, you will have more time to do this.
We should do this once a week, Vince, no matter what.
Well, if you're fishing, I'll come and bring my microphone.
No. I'll come next week, I promise.
We could do it on the bank of the river.
I promise. We've got to do trypanosomes next time, by the way.
Okay. You know, my wife worked on trypanosomes.
I do know that.
I've got one correction to make.
Oh, good.
Just listen to the tapeworm episodes. It's a megaloblastic anemia because vitamin B12 is needed for DNA synthesis and not for the hemoglobin.
What did I say it was?
I don't know.
I thought it did say megaloblastic.
Well, anyway.
Megaloblastic just means they're large, pale red cells.
Okay. Anyway, it's megaloblastic.
I meant to say megaloblastic.
And a question. Why the heck are those life cycles so complicated? Does anyone have an idea how they evolved? Maybe Dick has some good ideas
how they
on
evolved. You know, life in general is very complex. I mean, look at the life cycle of a coral reef, for instance, and look at how they reproduce. And I think the moment you start to look at biological systems, the complexity factor just jumps off the page. I mean, it's incredible.
Yeah.
So, I mean, viruses, in terms of their ability to take advantage of what's already there and re-engineer it into something else, I think that's fascinating stuff, and I think that's very complex. Maybe we'll just do a show on complexity once. I'd like to express my views on complexity sometime.
I think it's because you have multiple hosts and multiple cells within the host.
Well, as evolution occurs, and as these become simplified relationships, and then the organism repertoire increases, the possibilities for complexity increase.
Sure.
And by trial and error, nature works by trial and error. There's no question about it. And by numbers, sheer numbers of things.
Yeah.
I think by luck and by happenstance, we have to account for all of this by simple deductive reasoning to say, if this is the end result, then it must have arisen because of all of these other events prior to that. And so, I mean, you can't discount the fact that they exist, so therefore you have to go back in time and say, at some time way back, you know, during the Cenozoic era or so, or the Carboniferous era, these were simpler life cycles.
Right.
But as life forms became more complex, it allowed for the complexity of parasitism as well. That's not a great answer, but it's as good as I can do. Sorry.
Then he says, I plan on buying the Parasitic Diseases book.
Good.
But I noticed it's from 2005.
It is.
And the edition before was from 2000.
That's true.
So please be honest. Is there a new edition coming out soon? Please
Not
tell
soon.
me if it's so.
Okay, there's none coming out soon. Within a year. The answer is within a year. So if you want to wait a year, that's fine. But if you don't, I'll be happy to sell you this copy that's been selling for the same price for the last 7 years. We haven't raised it a nickel.
Thank you, Mark, from Germany.
Well,
the last one, I said this was the last one, but this is very short. It's very cool. This is from Michael. He sends us a website, which he says, can you believe this? There is an organization trying to save the guinea worm. Now I've heard it all.
They're trying to save it?
Yeah.
That's
defending
absurd.
the world's most endangered species.
That's got to be a joke.
It's a foundation. Who speaks for the guinea worms?
That's got to be a joke.
As you read this, a cartel of powerful organizations is conspiring to exterminate a living endangered species from the planet, and almost no one is doing anything about it. It must be a joke.
It's a clever twist on WHO.
Anyway, we'll put that in the links. Thank you for that, Mark. And that will do it. We'll say
that was a better joke than the Taniasolium joke, by the way.
Do you have a pick, Dick?
I do, actually. It's the new book out by Sarah Shah, S-H-A-H.
Ah, yes.
And it's on the history of malaria. And I confess I haven't read the book yet. I've just ordered it through Amazon, but I read the reviews and they're spectacular. And this is a Boston journalist that has taken the world's literature on malaria and amalgamated it in a way that hasn't been amalgamated before, because there's a tremendous number of books on the history of malaria. Mm-hmm. Apparently this one speaks right to the heart of the matter and gets to the nuggets of where it was discovered and how it was discovered and the incredible devastation that it has wreaked on the human population, and how back in our early history in Africa we almost became extinct as the result of it.
Cool. Maybe she talks about the spread from Africa as well.
Maybe.
All
Well,
right.
if she did, it's because someone else knew about it.
Yeah, sure, because she's reading the literature.
That's
yeah.
right, that's right.
Excellent.
But apparently she's done a wonderful job of translating that literature into a readable book.
That's a good pick, and I will buy it too, and
Great.
I will read it. Maybe
Great.
I will buy it.
No, you don't have to buy it, Vince. I
electronically.
bought it already. It's going to come, and
You're
we'll
going to
share
lend it to me? Good, we'll save a tree.
Yeah, we will.
Thank you, Dick.
Save a tree. Maybe we should get a Kindle and save a whole bunch of trees. Although I'm not sure about the carbon credits. If you look at how much electricity is used to run the Kindle, it's just it might come out to be the same. But did you know, by the way, that for the very first time in the publishing history of the world, more books were sold electronically to Kindles than hard copies?
At Amazon,
that occurred last month.
Yeah, that's very good.
That was an amazing statistic.
Well, I don't want to buy any more paper books because I have an iPad and I can either get Kindle or iBook books, but many are not available yet, including my textbook.
Well, my book that's about to come out in October on the Vertical Farm is both an audio book, a Kindle book, and a regular book.
That's great. And when it comes out, we will pick it.
Off the floor, probably.
No, not at all. We will make it a pick of the week.
How? That would be very self-serving.
Well, I'll do it so you don't have to.
Oh, okay. Thanks, Vince.
Well, if you're a new listener to TWiP, why don't you subscribe in iTunes? It's free, and it allows you to get automatically each new episode as we post it. If you don't use iTunes, you can always download the episodes at microbeworld.org/TWiP, where we also have show notes. If you like TWiP, leave a comment on iTunes. It really helps us stay on the front page of the Medicine Podcast directory, and write a little review. TWiP is also part of sciencepodcasters.org and promednetwork.com, websites where you can find high-quality science content. As always, send us your questions and comment to twip@twiv.tv. Good to see you again, Dickson.
Vince, it's a pleasure to be
here. Always fun to do podcasts with you.
I totally agree.
Dickson is at verticalfarm.org or .com and trichinella.org.
Or medicalecology.org.
Yeah, he's got a media empire. He's great. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.